for just accumulating mutations. Now even they would not accumulate all mutations, because anything that leads to outright sterility or mortality, you can't propagate that obviously, right? But at least you get the majority of mutations in there. That's the best way to estimate what a mutation rate is. So, and the theoretical underpinning for these, of course, is that and this is one of the most profound lessons for me in evolutionary biology, that the effect of a mutation and whether it goes to loss or fixation is not just dependent on its fitness effects, it also depends on the population size, right? So it's basically dependent on selection coefficients of the mutation, and most mutations tend to be negative or deleterious, so they often have negative selection coefficients, but also the NE. And the whole idea is that fate of mutations with selection coefficients less than 1/2NE, this is for a diploid outcrossing species, is governed by drift, whereas those with selection coefficients greater than 1/2NE is governed by natural selection. What this means is that mutations with intermediate to large selection coefficients, and again, in MA experiments we assume that most mutations are negative. So we don't even put the minus sign, but the idea is that they're deleterious to fitness. We know that pretty much, that most mutations do tend to be deleterious to fitness when they arise. So the idea is that mutations with intermediate to large selection coefficients, no problem, even at small population sizes, they would be eradicated via purifying selection, right? And if the population size is high, at high NE. So yeast should have no problem taking those out. Bacterial populations, no problem. But these mutations can behave in an effectively neutral fashion and reach fixation by drift at low NE. So a mutation may not be neutral with respect to fitness, but if it's in a population with a low effective population size, it can behave neutrally, accumulate in the genome. So that's the whole idea of these MA experiments. And here's just a schematic of the way they're done. You take an ancestral line, a homozygous line as much as possible, a single worm in our case, and you then get its descendants to make independent replicate lines. And then each of those is evolved independently over time. And as you can see, they start randomly accumulating mutations. So you see each subset of line, each line has its own set of mutations, presumably mostly deleterious. And the idea is you then go ahead and after letting sufficient number of generations pass, you go and assay them either via fitness assays, or you can nowadays just sequence the whole genome to get at what the mutation rate is and compare it to the ancestral line. This is what we did. So this was the first experiment I worked on. This was done in the Lynch lab when I was a grad student. It was the longest spontaneous mutation accumulation experiment. And we want to do it under spontaneous conditions. A lot of people try and accelerate the rate of mutations by using EMS or other mutagens, but it has a different spectrum of mutations. So we try and stay as close to the spontaneous spectrum, so we just let them accumulate naturally by just bottlenecking them. But that takes time. So this experiment was done over, it went on for at least, there were 10 people working on it, and it went on for 5 to 7 years. Every 4 days somebody moved the lines and set up a new generation. So we had about 400, 500 generations already. And of that subset, a lot of them had gone extinct, but essentially it was a single worm, hermaphrodite, and we had 100, I think it was 100 lines to begin with, and then they were each propagated. Every generation from each plate, one single worm was picked as the founder for the next generation. And we did this for probably more than 500 generations. So when I started my first lab, the whole idea was this power of natural selection is so diminished with the effective population size of 1 that pretty much any mutation that has a selection coefficient less than 1/2NE is going to accumulate in a neutral way. And so we went and took 10 of those lines, of the Lynch lines, and first tried to get an estimate of the spontaneous duplication rate. And we used array technology, which was first coming out in the early 2000s, and it's just a more recent high throughput way of looking at copy number changes. It's far better than traditional karyotyping because it can detect copy number changes up to 1 kb, whereas traditional karyotyping only gave you a resolution or copy number changes that were 5 million base pairs or higher. Nowadays, people prefer to bypass this. You just go and sequence the whole genome. That's far easier nowadays. But when we did this in early mid-2000, we basically, and the whole idea behind it is you take the test DNA and the reference DNA. So in our case, the test DNA would be our ancestral control, and the reference, our test DNA would be our MA lines after a certain amount of generations. The reference DNA is the ancestral control. You label them with 2 different dyes, and then you basically have a chip which has, the particular chip we used was the C. elegans from Wimbledon, and it had about 385,000 probes. We tried only use unique probes, and there were on average about every 75 nucleotides in the gene. And now you take the two fluorescently labeled DNAs and hybridize them together onto the chip, and they compete to hybridize. And the whole idea is that if they are in equal copy numbers, the test and the reference, you'll get a yellow signal, right? Whereas if there is a copy number change, if there's been a loss in the test DNA, a deletion in the test DNA relative to the reference DNA, you're going to get a higher signal for the reference DNA, more green fluorescent signal. Whereas if there is a duplication in the test DNA, you have a lot more DNA of that, you're going to get a stronger signal, fluorescent signal for the test DNA, signifying a duplication. And of course, you don't have to try and figure out if you have color blindness, etc. You don't have to try and squint at the colors to see what color they're giving you. You just have a scanner and software that reads the colors, the fluorescent signals on the chip after you do hybridization, and basically puts it as the output as a log2 ratio of the two strains. And that's the position of the chromosome. So this is the kind of graphs it generates. If anything gets to 0, essentially means equal amount of DNA in the copy, in the test, and the reference in the genomes. But if you have something closer to 1 or higher, it signifies a duplication in the test genome, and anything less than 1 or lower is -1 is a loss, a deletion event in the test gene. Everybody looking at this? Okay, so we just look at these plots then, and this is what we found. When we looked at duplications, we found 14 duplication events, but they weren't often just restricted to one gene. They often covered multiple genes, so they gave rise to span 30 loci. They led to 11 complete duplications and 19 partial. And when we look at the rate, if you look at the number of duplicated genes divided by the number of genes in the C. elegans genome, and how many generations we assayed across the lines, on average they've gone through 432 generations, MA generations, times 10, that's 10 lines, we got a rate of 3.4 times 10 to the minus 7. Remember, the bioinformatic estimate before that, which was considered to be astronomical, was 10 to the minus 8. So we've already gone order of magnitude higher rate than previously found, based on bioinformatic analysis. Now, we thought this rate was a downward biased estimate for several reasons. One, the probes we used were unique. Duplications tend to arise in repetitive regions. We had no probes for that region because they were repetitive DNA, so we didn't have probes. Similarly, when you have small duplications, there may not have enough adjacent probes to detect. So we probably will not, we have a detection bias against detecting small duplications. So we think that this very, very high rate now is actually a conservative estimate. But already this rate was about 2 orders of magnitude greater than the point mutation rate. Right now it's 10 to the minus 9 per nucleotide site per generation for the species. And it's also 2 orders of magnitude greater than Lynch and Conry's bioinformatic estimate when they looked at the distribution of duplicates in the genome. That discrepancy, that we see a lot more duplications here, tells us that the average effect of a gene duplication is slightly deleterious. There's a whole bunch being weeded out before we even see them. We don't see them in genomes of the selection, but we can see them in experiments. Okay? We also did the rate of gene deletion. We had 11 deletion events that encompassed 19 complete or partial open rating frames, and the rate is about 2.2 times 10 to the minus 7 deletions per gene per generation. This is not deletion of the duplicate. This is just a deletion of a particular gene. And this is still pretty high, but it's still 1.5-fold lower than the gene duplication rate. And that makes sense. You would think that deletion of a single copy gene is going to be more deleterious than deletion or duplication of the gene. So this is not surprising. Then we looked at the span of these duplications versus deletions. If you look at the graph initially, you would think the deletions are much more frequent, but actually larger. The span is higher. The range of deletion events we saw was much higher. But the median deletion span was only about 3.5 kb, but the median duplication span in these lines was about 7 kb. Okay? So it's significantly, the deletion span was significantly smaller than the duplication span. And in these lines, in these 10 lines, we had a net increase of about 26 kb via duplication. 106 kb got duplicated, but 80 kb got deleted. So we surmise that because the deletion rate is lower, that some deletions are so detrimental they lead to outright mortality or sterility. We can't accumulate those mutations at all. They are more deleterious in their fitness effects relative to duplications. But there was a net accrual in genome size in these lines. So the conclusions we have is that the spontaneous rate is higher, way higher than we imagined. It's 2 orders of magnitude 200-fold greater than the nucleotide substitution rate per site for the species. This high rate is also 2 orders of magnitude greater than what was previously determined from bioinformatic analysis. Just looking at the extent of duplicates in the draft genome that was under selection, which tells us that many gene duplicates are efficiently purged from the genome by purifying natural selection in the infancy. You won't even see them when you sequence it because they just weed it out. But this high rate then contributes pretty much any species you look at now, you have astounding levels of copy number variation. And this high rate of input is then what's contributing to the huge CNV copy number variation that you're seeing in population genomic studies. Any questions so far? Okay. So then, we know right now from this, we can see there's a slight discrepancy in the rate, so maybe gene duplication is slightly deleterious. But we wanted to do a better test of this. So we were trying to now get at what is the average fitness effect of gene duplication. And this is done with Don Merman, who was at UBC, and he runs the C. elegans knockout consortium, and pretty much really developed the array technology for detecting duplications and deletions in C. elegans. And we're also working with Bob Waterston, who's at UW. He did the whole genome sequencing of our lines that I'll talk about in a second. So this is a new MA experiment that we began in my lab when I established it in 2006 in New Mexico. We did an MA experiment again, but with a twist. In this case, what we did was, rather the traditional MA experiment that tries to bottleneck the particular species, populations of the species, the minimum effective population size. Drosophila, 2 individuals, male, female, or rather sister pair, C. elegans, a single individual. What we did was we did the first spontaneous MA experiment at 3 different population sizes. Okay? We did it at size 1. We had about 20 replicas. 20 lines were maintained at size 1 every generation, 1 hermaphrodite. 10 lines were maintained at size 10. 10 individuals moved every generation, and the 5 lines, we just logistically couldn't do more. We had 5 lines picked by hand, 100 generations each individual. This meant just this simple design. We had 35 MA lines across 3 population-sized treatments, meant we were picking 625 worms every 4 days for 5 years. Okay? And so that was logistically all we could do in terms of sample size to create these lines. And so the whole idea is by doing this, and then the ancestor was, they were all descended from a single hermaphrodite, which was frozen as ancestral control. So what we were trying to do was now, by doing these different population-sized treatments, we are tweaking the power, amount of natural selection versus drift operating in each of these 3 treatments. Okay? And so the whole idea is, so that's what I just said, we're tweaking, it allows us to manipulate the strength of natural selection versus genetic drift across these different population-sized treatments, and now it enables the subdivision of the spectrum of mutational effects into successively narrow classes. That's very fancy verbiage. What do I mean by that? Here's what I mean. So that's based on theory. So if at size 1, you can see, you're going to accumulate mutations that have, and all these selection coefficients we assume are negative. Okay? So we're going to accumulate mutations in the size 1 lines, everything that leads to 50% decline or less, we're going to accumulate at the neutral rate. Okay? At size 10, the effective population size is 5 because of the partial inbreeding system of the species, but we're going to accumulate mutations that lead to 10% or less decline in fitness. And at size 10, selection is the most efficient in this background. We're going to only accumulate mutations that are close to having very low selection coefficients, 1% or less. This is important. Is everybody with me? So now we've got different levels of selection and drift. Drift is strongest in size 1, selection is strongest in the size 100 lines. And now we can compare the mutation frequencies for any class of mutations. We're right now focusing on just duplication deletions. We can compare them to see how they're accumulating and get to know what is the average effect, fitness effect of these duplications. So here's what we did. We did array CGH on this set of lines after 409 MA generations. And we look at frequency of duplications and deletions of these different treatments. I'm only going to present the duplication data, and here's our predictions. If there's no significant difference in the frequencies of duplication and deletions between these 3 population-sized treatments, the whole idea is that they're accumulating, they're neutral with respect to fitness. Right? If you have the same amount of duplications at size 1 and size 100, they're pretty much they're almost neutral. But if you see a lot more duplications or deletions at the low population size treatments, size 1, compared to the higher population size treatments, it tells you that they're deleterious with respect to fitness on average. And vice versa, if you see far more duplication deletions at high population size, it tells you they're beneficial to fitness. Everybody with me on this? Okay, so here's what we did. We looked at these lines after 409 generations, and here's what we get. We had one line go extinct, so we only had 19 out of 20 lines to look at for the size 1 treatment. So we have size 1, 10, and 100, and here is the duplication rate. So 6.9 times 10 to the minus 6, we have a new, firstly, again, before I go further, this is a new estimate we have. Remember the first estimate from the previous M experiments we did, we had a duplication rate 10 minus 7, which was 2 orders of magnitude from the bioinformatic estimate. We've gone 1 order of magnitude higher. Why? We used a different chip design. We allowed twofold redundancy. We had, in the previous chips, we had 385,000 probes, now we had 720,000, and sure enough, we went an order of magnitude higher the duplication rate, as we expected. So, we have a newer estimate, now it's 6.9 times 10 minus 6, which is huge. This is the highest estimate now for any species that's been tested, and it's twofold higher than the yeast duplication rate. It's also 20-fold higher than our previous estimate, and it's 3 orders of magnitude higher than for mutation rates. So we expect a lot of genetic variation being created by gene duplication. Now let's compare the rates with this larger population size treatments. Now that's in the next graph. Here, I've just put log population size in terms of log values. So it's size 0 log is, of course, population size 1, 10, and 100. And that's the log duplication rate across these different lines. We've done both parametric and non-parametric tests, and we find a significant difference. A much, much higher duplication rate at size 1 than at the larger population size treatments. And it's 6-fold greater duplication rate at size 1 relative to the higher population size treatments, which tells us that on average, a gene duplication, even though we know all these examples of novel traits arising from gene duplication, on average it's deleterious in fitness, and it's being weeded out at the larger population sizes where selection is more efficient. We also looked at the median span of these duplications occurring in these population size treatments. So again, the median duplication span in size 1 was about 5.5 kb, but much higher in to larger population size treatments, more about 9 to 9.5 kb at larger population sizes. So again, a significant difference between the in the span of the duplication in the 3 population sizes, which tells us that larger populations which have much larger duplication spans. Okay? So smaller gene duplication somehow, maybe they are mostly partial duplicates, they are also being weeded out at larger population size. You don't see. So at larger population sizes, lower duplication rate, but much more is being preserved. Okay? So higher duplication rate, but smaller duplication spans in the smaller size treatments. So just in conclusion, the more sophisticated chip design allowed us to get a higher probe density, which gave us a much higher rate. So now we are way beyond astronomical, I guess. We are at 6.9 times 10 to the minus 6 duplications per gene per generation. So this is the fact that it's significantly higher duplication frequency at very low population size tells us that the average fitness effect of a gene duplication when it first arises. The average, I'm not saying they're not good gene duplications, but an average when it comes out is deleterious to fitness. Okay? But we have a significantly larger span at higher population sizes, which tells us that even partial, small, attenuated duplication spans also are deleterious with respect to fitness. And so the third experiment I'll talk about, this is another mutation accumulation experiment because we love punishment. So the question was, are copy number variants a common form of genetic change during adaptive evolution? Again, this is done in collaboration with the Mormon lab at UBC. I think I've had problems with this slide.
It's even better the second time.
All right, here we go again. to a slide. I wonder if you can specify a slide, if I can just bypass that all the way. I fear it's going to be this.